So there's two types of mechanisms that we usually have with herb-drug interactions. We have a pharmacodynamic interaction and a pharmacokinetic interaction. So pharmacodynamic is the herb causes additive, synergistic, and antagonistic activity in relation to the drug. Pharmacokinetic changes the absorption, distribution, metabolism, protein binding, or excretion of that particular drug. So both of these can happen simultaneously, and we all know this type of stuff. So again, this is a review. So pharmacodynamic interactions. So this is additive, synergistic, antagonistic. We'll give some examples. So you use ginkgo. This is an interesting article. Is there a bleeding association with just using ginkgo for average patients? And pretty much not really. If you just use ginkgo as a botanical and they're not on any pharmaceuticals, generally there's not a lot of risk of bleeding. But now throw in a couple of medications in there, and now it's a disaster. Now it's a big disaster, right? Combine it with RX or OTCs, totally different matter. And this is a big problem because a lot of people use these botanicals. They come in your practice, and they may not know this. So that's kind of like, an example of a synergistic interaction. You can even look at it on WebMD, and that's a screenshot of WebMD, and what you come up with is, to watch it when you use those particular things in general. Now, another great website is exam.com. That's free, and that's a screenshot of what it looks like. And I put berberine in there just to show you. Berberine is from many different plants, Chinese herbs. We find it in golden seal and a lot of other of those Ranunculaceae family botanicals. And you get some great information on here. Particularly, this is pretty interesting that, of course, it interacts with a lot of diabetic medication. And there's even some serious cardiac things that we'll get to in a bit that it could cause berberine. So there's certain things that you got to watch out, particularly the microlide antibiotics. If you combine with those things, it can actually lead to serious cardiotoxicity. So that's Z-Pak, I believe, right? Azithromycin. So Z-Pak, so a lot of people use Z-Pak and golden seal. They're like, oh yeah, well, I'm going to use the two together for an infection. That may not be a good thing if a patient has some particular heart problems. So that's one to watch, I think. So that's examine.com. Now, the great one that we use at school, and you could subscribe to this, but it costs a lot of money, is, of course, Natural Medicines, formerly Natural Standards. So that's what it looks like when you go online there. It's my secret Bridgeport account. So, and I put in the drug Asifex, just for kicks, looking for nutrient depletions, kind of backtrailing about nutrient depletions. This is how they list it, because you can do nutrient depletions on natural medicines, you can do effectiveness checkers, adverse effects, and then, of course, drug-herb interactions. So this is a really good one, but it's expensive to subscribe to. So you can see that Asifex, the major thing is depletion. Dr. Saunders just spent a lot of time telling about how magnesium is really important for us. And here you're taking something for stomach problems to reduce stomach acid and getting major problems with magnesium. There's dibengazide. By the way, what's dibengazide? Anyone know what that is? Dibengazide is a supplement that has 3 out of the 4 B12s all in one, I believe. You can Google and just fact check me on that. But I believe it has 3 out of the 4 B12s. So, that's kind of interesting. But you could see that they rate whether it's a major depletion, minor depletion, and then if you follow up, they have the greens, which is like insignificant depletion. So I usually don't pay attention much to the insignificant ones, but if there's something red there, I'm going to read the literature on that and follow up on that just to make sure, because again, it doesn't all sometimes come out in the wash, whether it's an in vivo thing or is it based on a real clinical observation and problems that have been reported to the NIH when there are serious drug adverse effects and nutrient depletions that are reported to NIH, that's what I pay more attention to. That's what you're going to find in the red here. So in my opinion, that's what I'm really interested in there. So that's Natural Medicines, Inc. So you would go on Natural Medicines and you click interactions with drugs and supplements there, and you get, the interactions with berberine and antiplatelet drugs, anti-diabetes drugs, antihypertensive. So anti-diabetes drugs, big red flag. Berberine is very effective at lowering blood sugar. We've known that for centuries. And you may need to either lower people's various drugs, and they give you the list of medications that it interacts with there too. And there's a little art, and that's a hyperlink, treatment of type 2 diabetes and dyslipidemia with natural plant alkaloid berberine. So we'll just continue. And then they do herb-to-herb interactions too, which I think is cool, natural standards. Like what herbs can you mix with other herbs and which herbs can you not? So that's very valuable, especially if if you never made a tincture before in your life and you don't know what goes with what. This could be very valuable. So back to our herb-drug interactions. Antagonistic reactions would be, of course, like taking Xanax and guarana, or kava and coffee tonight. So you could see that they just canceled each other out, basically, and that's what basically that says. So that's an antagonistic interaction. And then synergistic ones would be like taking metformin with gymnema or metformin with the berberine, like we talked about, and you're going to get a synergistic lowering of blood sugar. And it's going to be additive is 1 + 1 is 2, synergistic is 1 + 1 = 5. So it's going to magnify the effect when you take berberine and many of your diabetic drugs. So much so that they could just go into hypoglycemic shock very quickly. So we'll skip that one. So now we want to spend most of the time on pharmacodynamics because that's really where all the interesting stuff is there. And of course, we know a lot about cytochrome P450 systems and the CYPs. So a good example is ginkgo decreases the effectiveness of Xanax. But they found out that it actually decreases the absorption rather than inducing a pharmacokinetic hepatic metabolism. So originally, the researchers all jumped up and down, ginkgo's no good for the liver when you combine it with Xanax, blah, 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 but it really just have to do with an absorption thing. And that may be because of the tannins and flavonoids and the ginkgo flavoheterosides found in ginkgo may interfere with the absorption of things. You'll find green tea, by the way, at the end of the lecture. I have an article on that. Never take your medicines with green tea. Yeah, we'll see. So Dr. Gurley was the one who found that at the CYP level, CYP level, ginkgo really didn't have a lot of interactions. So it's more of an additive effect, but not really at the CYP level for particularly ginkgo. So we know about the drug metabolism pathways, and we know CYP is an abbreviation for, of course, cytochrome P450 systems. They oxidize. These are some of the chemicals that are used, or the detoxification pathways that the CYPs use there. Basically, the idea is to just make your drugs more polar and urinate them out, or they come out through the bowels or other ways. Most drugs metabolized by that CYP3A4, that's the big one, 40 to 50%. So we want to pay attention to that. Also, the 2DS and the 2C9. So that's a nice pie graph that I have usually by my desk so I can look up these different drugs and find out how they're metabolized. Now, of course, it's real funny when grapefruit came out and shown that it inhibits this CYP3A4 and caused an increased bioavailability of many drugs. A lot of my students would take advantage of that. They drink one coffee and eat a lot of grapefruits, and that coffee would give them a buzz all through finals. Isn't that really using their knowledge for good purposes instead of evil? I think it's great. That means that they're paying attention, right? Of course, the culprits in grapefruit are the furanocoumarins. These are the things that seem to interfere with the CYPs, and you get the increased level of concentration into the bloodstream. So that can be bad news, of course. Now, the Dietary Supplement Health Education of 1976 happened, and so many more people started to use herb and drugs, so the feds started getting more and more reports of herb-drug interactions. And it's interesting, this is an article that cancer doctors really don't discuss herbs with their patients because they don't know as much about them. So anyway, and they probably should. So hopefully with this lecture and your own personal studies and your practice, you'll discuss more of these things with your patients, especially the herb-drug interactions. That's what I'm looking for. So this was like one of the first articles of Journal of Clinical Anesthesiology where they found out that certain things seem to interfere with anesthesia, and that's something you don't want to monkey with, you know? If you're on a table and then all of a sudden you wake up and he's got the knife or laser above you, not a good thing. Not a good thing. So I pay attention to case reports, adverse event reports, and other clinical studies, but I don't really, the literature and particularly a lot of the stuff on WebMD and things like that, a lot of it is in vitro studies and things like that. I don't pay attention much to those interactions. I want to find out the adverse reports because that's when you may run into trouble. So of course, when we look at pharmacokinetic mechanisms, we have the CYPs, we have the glucosyltransferases, glutathione transferases, and the sulfatases. These are all how drugs are metabolized, of course, in the small intestine and liver. So that the CYPs are going to affect those types of things there. But also we want to talk about those p-glycoproteins and other ones that we've mentioned before, and we're going to go over more details of what they actually are. So some quick definitions: p-glycoprotein and OATP. So there's two major transporter proteins. You have efflux pumps and the uptake pumps, right? So the p-glycoprotein is an efflux pump. Idea is to pump it out of the cell. Or bacteria, pump it out of the bacteria. Pump that antibiotic out so that they can still live a life. Then you have your uptake pumps, your organic anion transport polypeptides, and cations too. So these are the ones that are going to either, put the drug in, take it out, and very important, if the herb affects these particular systems, that's going to be critical. And that's a picture of what's actually going on as these glycopumps efflux things. So we know back again that these CYPs are very important, and what we have to pay attention to the P3A4 again, just to review. And if we have a botanical that augments these CYPs, it could be a problem because less of the drug reaches the general circulation. So Dr. Gurley, the head NIH researcher on this, what he looked at is this particular example when St. John's wort became the poster child of highly inducing botanical agents for these particular systems. And this is really where what hit the fan when St. John's wort happened. But what's the story behind that? Anyone know the story behind how this was discovered? Dr. Saunders knows. So we'll skip that one. Now, I want to skip that one too. Actually, I'll go back. I just don't want to run out of time. So these are the particular substances that are unique to St. John's wort that interact with drugs. And what's interesting is when we look at most St. John's wort labels, we see the hypericin content and pseudohypericin, but we don't see the hyperforin content, and we don't see them standardized to those particular compounds. And that's because it's very expensive to do that. But those are the ones that are the troublemakers in St. John's wort. So this is an important slide because I pointed out what's the troublemakers. So we know that basically St. John's wort makes prescription drugs ineffective, many of them. So this was a case that Dr. Gurley, there was a woman with kidney transplantation taking cyclosporine. We know that it's metabolized by CYP and p-glycoproteins. Now, it turns out that it was like before Christmas, and she started taking St. John's wort for seasonal affective disorder without telling her doctor. And it was a smart nurse kudos to nurses, that picked this up, and they found out that she was actually starting to reject her kidneys, right? That it was interfering with the cyclosporine. And I'll show you the graph. And then these were some articles that came out in Archives of Surgery. Warn your patients getting organ transplants about St. John's wort. This is all historical stuff. We all know this already now. So this was the graph that Dr. Gurley wrote in his major paper in Transplantation in 2001. Look at the time on Hypericum and look at what happened to her cyclosporine levels. And she actually rejected the kidney and lost it. So this became big news. This is how we all found out about this stuff, you know. So, and it turns out that St. John's wort is one of the most effective detoxification herbs, and I actually have used this information to help detoxify patients. I purposely give them St. John's wort because it's such a powerful liver detoxer, but only when I'm detoxifying them and only when we're monitoring their drugs. I'll talk about that in a bit. How does St. John's wort work? Well, St. John's wort gets brought into the cell, and it binds to these things called the PXRs. I'll talk about that in a bit in the next slide there. PXR is an orphan nuclear receptor, which is located in the cytoplasm. And then it's brought into the nucleus, and that's where we have the gene transcription. This is all review for you guys. And then the St. John's wort will then inactivate things, you know. So that's kind of a cool little diagram. By the way, that's a good list of drugs there that St. John's wort really, really interacts with, you know. So you can see if you have a patient with HIV and they're on some antivirals, that can be a big problem, you know. And of course, St. John's wort is what? It's antiviral against HIV. So it's like either or. You better either do the St. John's wort or do your antiretrovirals, but don't do them together. And a lot of people do, and they run into problems. This is the major domain where the St. John's wort attaches, right here. This is the nuclear receptor right here. And it attaches to this ligand right here. And then they found out that Hypericum is the most potent ligand ever discovered. It virtually just turns this on hugely, the CYP systems, and it's the most powerful inducer of those efflex pumps, those it's supposed to be p-glycoprotein. Apologize for the typo. P-glycoprotein pumps. So you got problems with, and this is about the p-glycoprotein pumps, but and they're part of what they call ATP binding cassette superfamily of transport proteins for those real cellular kind of nerd people that like this stuff like me. Very nerdy. And just examples of what's happening with the, again, the p-glycoproteins there. That's the cassette binding. So you get actually a conformational change, and boom. So these are very important things that we know that St. John's wort interferes with here. So amitriptyline, digoxin, that's kind of scary, and some other ones there too. And again, we talked about the standardization of hypericin versus the hyperforin. So it's hard to do that. Again, more drugs. We keep finding new drugs there. Now, again, I talked about that I've used Hypericum for detoxing people. Now, you want to support phase 2, but use the Hypericum to upregulate phase 1, and you could detoxify a lot of those pops that Dr. Pizzorno was talking about Thursday.